Okay, thanks very much. Welcome back to whatever this is, ISC 6930. And this is the last class for the semester. So there will be no classes after this. That's what defines this as being the last class. So what we're going to do today, which we've never done before, is pick up where we left off. So if we go back to the Elmo. Thanks. And get a little wider. What we've been doing before is we've had, first of all, this is the usual list. We've had these Gaussian statistics, which are run through faster than usual. Gaussian statistics, and then non-parametric statistics. So the Gaussian ones where we had a nice defined mean and variance that parameterizes the probability density function. Here we have probability density functions where we don't need to know the mean and the variance. And then we got into these fractals where we have no probability density function or a non-normalizable one, and where there are no mean and variance. What we've been discussing since last time is chaos, which is different than all of these, because in all of the previous ones, we've been assuming that we have, so before, we had basically random, small, numerous errors. And then what we're learning in terms of chaos is that we can have systems that look random, but they're not random. And so far I've gotten through one page with no spelling errors, but I'm sure that will change. So what I want to do today is to continue with these properties of chaos, which what I started to define last time, and go more into what the mathematical properties are and illustrate them with biological examples. One of the things I mentioned last time is that if anyone had any data sets that they were interested in analyzing that I could, say anything useful about them, they should bring them in, but nobody brought them in, so we'll presume that nobody has them right now with them, so we'll not do that, unless someone comes in holding a large sheaf of paper with data on it. So I'm going to continue. Are there any outstanding questions from last time on the properties of chaos that Okay, that's all right. So where we left off last time, or not quite where we left off, but to backtrack a little bit to set up what we were doing, was we had the defining properties of chaos, I think, are three. First of all, it's deterministic. So we have the next value can be predicted by a few previous values. So in this case, we have these three previous values, then predict the next value of the system. And the second property is that there are only a small number of variables that do this. So in this case, as I said, the 3 previous values predict the next value. And the output of this system is very complicated. In fact, it's so complicated it looks random, even though it's not random. And the properties of chaos that I've been describing are, first of all, that we can transform the time series into a phase space. And as I said, Poincaré did this about 100 years ago because there are properties of the system that are more easy to learn about in terms of the phase space. And in this case, we can see that the time series that produced this phase space fills it up, and so it was more likely to be random. But the time series that produced this phase space is one-dimensional. It's this one-dimensional object called a strange attractor. So that this time series is really deterministic and chaotic, and this is stochastic and random. And the second property, which we'll discuss a little bit more today, is sensitivity to initial condition, unless we discussed it last time and I forgot, is sensitivity to initial conditions. So if we start off somewhere with 2 points that have nearly identical initial values, as the system evolves in time, those 2 points will evolve to very different values. And the third property of chaos was bifurcations. And this is the point which we ended at last time, that we can have one pattern. So this could represent different values in time, the dots representing different measurements, which are then connected by a straight line. And you can see here we have one pattern, but when we change a parameter in the system by a small amount, we get a very different pattern. So we would call this periodic and this chaotic. And this is very counter to what our usual assumptions on things are. We're really used to thinking that if we change things in a small way in the system, we'll get a small change in the output. And here we change things in a small way, and we get a huge change in the output. And so this is an example of a bifurcation. And let me now scroll forward to this was sensitivity of initial conditions that we did discuss last time. Okay, this is where we left off last time. So let me give you some real-life biological examples. And this is not quite real life. This is in a test tube. But it used to be alive. These were cells that were ground up, and their enzymes are still working. This actually raises an interesting issue. It's not clear if there has ever been an example that people have noticed directly in an in vivo system that has chaotic properties. So there are certainly examples of systems derived from living systems that have this, but what role this plays in cells or in real systems is not clear, and there's a lot of debate on this right now. So maybe it's interesting that this is done in a test tube. And what this is glycolysis. That is, energy in glucose is transformed into adenosine triphosphate. And ATP is important because then the energy stored in the high-energy phosphate bond in ATP is used as an energy source to drive many other biochemical reactions in the cell. And this shows in a simplified diagram the biochemical pathway of the ATP. So we put in a sugar at this end, which is fructose 6-phosphate, to show that I know what the little letters stand for. But it's basically a sugar, and then that sugar is converted ultimately into ATP. Now, what's interesting about this pathway, which is somewhat simplified here, and in fact, this is the pathway that does not use oxygen. There's a more efficient way of producing ATP that does use oxygen. But the rate of these two rate-limiting enzymes, and I know what these letters stand for, but not those, so I shouldn't say what these are, because then I'll come over here and get stuck on these. But this is phosphofructokinase, and this one I don't know. But the point here is that these are the two enzymes that form the rate-limiting pathways in this series of chemical reactions. And what's interesting about these enzymes is that the concentration of the products, in this case, have a negative feedback rate on how fast the enzyme works. And in this case, on this enzyme, the rate at which this enzyme catalyzes this chemical reaction depends positively on the concentration of this product, ADP, and depends inversely on PEP over here. So this means that the rate at which this reaction is happening has positive feedback from this product and negative feedback from this product. So this is a very complicated system. And this is one way to get chaos, by having negative and positive feedbacks on a system. Now, this reaction was studied by Marcus and Hess in Germany. They used to be at Dortmund. I think one of them moved someplace else now. I don't remember where they are now. And what they did is they wrote down the, get back here for a moment. They wrote down the set of ordinary, nonlinear differential equations that represent what's going on in this process. So maybe if I can just give a hint of what these equations look like. That is, they're going to say that the rate of change of ATP concentration in time depends on some factor times the ATP concentration times some different factor times the ADP concentration, etc. So they can write down a differential equation. Now this is a nonlinear equation because this squiggly thing, you might have thought it's a Greek letter, but it's not. It's just a squiggly thing. Actually depends on ATP concentration and ADP concentration and the glucose concentration being put in and lots of other things. Now, where did they get these dependencies? And the answer, they got those dependencies that were measured experimentally. So there are experimental values of how these coefficients depend in a nonlinear way on all these concentrations. They're very complicated relationships. In the fractal and chaos course where I describe this in greater depth, we'll see that these depend on like temperature to the 6th power and ATP concentration to the 7th power and all kinds of funny things. So these are very nonlinear. So we have a set of nonlinear coupled, because the ATP and ADP are linked together, ordinary, meaning they're not partial, first order, because although they're complicated algebraically, they only have one derivative with respect to time, equations. And so they could write down these equations and solve them in one of the usual ways. And my usual way for solving equations like this would be fourth order Runge-Kutta integration, which is not considered very fancy, but I think is effective. So if we go back here, and this is what they found. If they put, in this case in the actual experiments, they didn't put fructose 6-phosphate, they actually put glucose into the system. So I'll talk in terms of either glucose or sugar. If the input into the system, that is, the input into this end over here, into this funnel at the beginning of the system, so they're going to put stuff in here, drip stuff into here. And these are in test tubes. They're in a glass test tube. They've taken these yeast cells, which I think is brewer's yeast, but I'm not sure. And they've ground up the cells, so the cells are dead, but all their biochemistry is working. They have all these enzymes. And then they literally have something dripping into the test tube, which is the sugar input. They're measuring ATP because there's a fluorescence technique where they shine in ultraviolet, and the amount of fluorescence depends on the ATP. More accurately, it's actually the NADH fluorescence. So that's what they're measuring. And I don't have any pictures of that. So here, from the theory, from those nonlinear equations I hinted at, if they put in the sugar in an oscillatory way, they predicted that the output of ATP would also be oscillatory with the same period. It might be phase shifted with respect to this, but it's periodic. On the other hand, you can see here they predicted if they increased the frequency at which they put sugar in a little bit, then they predict that the output should be chaotic and not be periodic, even though the input is periodic. And this is what we've seen before in terms of the beating of the heart cells, if you remember, where the heart cells were stimulated at a regular rate, and yet they beat at an irregular rate. Okay. So let me show you their actual results of the experiments. So they've used baker's yeast. They're measuring ATP by a fluorescence technique. And in each of these plots, the ATP measured will be on the top, and the glucose input, which is they'll be putting into the system, will be on the bottom. And the horizontal axes will be time. And this is actual experimental data. So you can see here when they put in the glucose at this oscillatory way, they get an oscillation in the ATP. Now you can see that the the peak of the glucose does not correspond in time, in phase, to the peak of the ATP, but they have the same period. Now look what happens when they put in the ATP, they put in the glucose at a little bit faster frequency. And now here, they're putting it in a faster frequency, and this is the output of the ATP measured by the system. And this is really pretty astounding. I mean, you can imagine what would happen if you got a result like this and you tried to publish it, or you tried to write a grant to do more of this. I mean, the first impulse would be to say that there's something broken, that the sensor that you're using to measure ATP isn't recording things accurately, or you're not doing the measurement properly. And the second impulse might be to say that the reason why this is fluctuating is something isn't being held constant in the system. That what's happening is maybe you're not holding the temperature or the pH or something is changing in the system. And I want to emphasize that these fluctuations have nothing to do with experimental error, and they have nothing to do with the system not being held constantly. That these fluctuations are really the essence of what a chaotic system does when it's chaotic. Now, when I say this, I always ask, does this mean that every time we see data that looks like this and has fluctuations that it's due to chaos? And the answer is, of course, no, because we can easily have broken sensors and we cannot be keeping things constant in the experiment. But what we have, and what I've been developing here in this lecture, is we have tools to analyze this to tell whether it's really chaotic or not. We've seen that one of the tools would be to embed this in a space and then measure its fractal dimension. And if the dimension was low, we would know it's deterministic. And if the dimension was high, we might think that these fluctuations were chaotic, were random. And in fact, they did that to this signal. I don't have a picture of it here, but instead I have a picture of another technique that they used to analyze it. Now, this shows both the computations from the theory compared to the experiment. And what's being plotted here is on this axis, on the horizontal axis, is the frequency at which they're putting glucose into the system. So here they're putting in at low frequency, so it takes a long time for the glucose to go through the cycle. And here they're putting in the glucose at high frequency, so it goes through the cycle in a short time. Okay? And what's being plotted here is they're plotting, if you could switch to the camera for a moment. Hi guys, if you could switch to the camera? Maybe. So instead of doing that, we'll do it here. Basically, what they're doing is they're putting in glucose in an oscillatory way. In fact, this is probably better than waving my hands. So we're putting in the glucose like this. And we're measuring the ATP. So this is time. Time feels blue today. And we're measuring the glucose at the same phase each time in the cycle. So here, I just picked one particular point in the cycle, but we want to measure the glucose, the ATP, at the same phase. So at each of these points in time, we're going to measure the ATP level. Now, if the ATP was really moving with the same period as the glucose, even if it moved out of phase, we'd see the same value of the ATP each time if we measure it at the same phase of the glucose cycle. Okay? And let's see what we find in the data. It's not as clear, but it's very dramatic when I do this with waving the arms, actually. Maybe just dramatic, but not very dramatic. So in this case, the theory predicts from the set of ordinary, coupled, nonlinear first-order differential equations that for this given frequency, we have one value of the ATP concentration. So that means each time in the cycle, we get the same value of ATP. Now, as we increase the frequency at which we're putting in ATP, suddenly over here we get a change in behavior, a bifurcation. So this is a bifurcation diagram. And now we have two values of ATP. So that means again, if we're looking at the glucose and we measure at the same point in the glucose cycle, it means, let's get some more cycles here. And now we look at the ATP. It means one time we have a high value for the ATP and another time a low value. And the next time a high and the next time a low and then a high and then a low. So if you look at this, the ATP is going through a cycle, but it's going through a cycle that takes twice as long, okay, as the glucose cycle. So if the glucose cycle takes time capital T, the ATP cycle is taking 2T. It takes twice as long. And this is called a period doubling. And since there's a sharp change of behavior, it's a period doubling bifurcation. And this is very typical of chaotic systems. If you've had Ming-Shu Ding's course or will have had Ming-Shu Ding's course or have had my course in chaos, this is one of several types of bifurcations that we can have, or the glucose can have. So here we have a period doubling bifurcation. And then here we have even more bifurcations. We have many more periods. And then look what happens when we get into this regime. Now the values of the ATP are spread all over the place. Well, how can that happen? What does that mean? So again, if we have glucose, the frequency is a little bit higher here, and we're always looking at the same phase. If we have different values of the ATP, it means it's all over the place here. And so this is really what chaos looks like. All right? So when we have the values spread out in that bifurcation diagram, we're really within a chaotic regime. And that's what we have over here. And then in the chaotic regime, there's this window here of period 1, 2, 3, 4, 5. We get back into chaos again, and then back to a period 3. So this is what is predicted by the theory. Now, on the bottom here was what was actually measured in the experiment. It may be a little bit hard for you to read, but the numbers here say 1, 1, 1, and then they start saying 2, meaning there are 2 values. We have a period doubling. Then it goes up to 7 here. There's a C here for chaos, and then the window 5, and then chaos, and again, and then 3. So although there's a shift a little bit in the parameters, in the parameter of the frequency compared to where the bifurcation sequence was found and where it was predicted, the experiment runs through all the different behaviors in the proper order as what was predicted. And this is absolutely amazing to me. And it shows that we really understand this system, that the equations are really predicting this very nonlinear set of bifurcations, and all of them have been seen in the right sequence in the experimental data. And that's really phenomenal to me. And I think this is a good way of showing that we really understand what the system is doing by having the system change when we change a control parameter, which in this case is the frequency at which glucose is being put in, and seeing actually those changes. So this is a nice way to do it. In fact, this is the paradigm that Kelso here uses, where you have a system and you change a parameter, and if you think you understand the system, you should be able to predict the bifurcations or the changes that the parameter goes through. And when you actually see those, it gives you some confidence you really understand the system. That just says what I said. And speaking of Kelso, what's come up here is an example of that. So here we have an experiment where we have a sound or a light. In this case, it's actually a sound. It's a tick of a metronome. It's probably not a real metronome. It's an electronic metronome. I don't know if they ever did this with real metronomes. No? No? No. Real metronomes are sort of interesting because they're inverted pendulums. So you change a little weight, and it changes the frequency at which it's moving. So they're sort of interesting. But that's mechanical technology. That's in the old days, when they made things out of wood and metal instead of plastic, and built buildings out of stone. So here the deal is we're tapping one finger in phase with the tick of the metronome. And what you're trying to do is tap the other finger out of phase with the metronome. So if you could switch to the camera. Thank you. What we're doing, and I'll use these, maybe here. Thank you. So somehow I don't think I should hit the electronics too hard. So you're doing something like this, and then as you increase the frequency at which you're trying to do this, you wind up tapping them both together. So I'll do it again. So you can hear the metronome. I can hear it. So, and as the metronome is speeded up, as I continue to try to do this, you can see I stop eventually, and both things are tapping together. So thanks. If you go back to the computer, the Elmo. Thanks. And as the frequency of the metronome increases, the right finger suddenly shifts from out of phase to in phase. That is, both fingers are doing the same thing. And this shows an example of data recorded from such an experiment. So one is the motion of the right finger, and the other is the motion of the left finger. These are green and something. Well, doesn't matter. Basically, they're out of phase here, and then as the frequency increases, they move to being in phase here. And this shows the relative phase of the two fingers. So here they're 180 degrees apart, they're out of phase, and then they move to being in phase. And the point of this is that this shift, this bifurcation, has many of the properties of a phase transition. That is, the whole point of this experiment was that Hermann Haken had shown in certain physical situations in lasers, and lasers are, of course, an acronym for light amplification by stimulated emission of radiation. And he basically showed that a system that had many pieces could sometimes act in a very simple way, as if it had few pieces. And he called this self-organization, or synergetics. And this is the situation that we have in the body. In many different situations, what Scott Kelso was showing is that, for example, if you reach for something or you observe something, so the sensory and motor system in the body are always doing things like this. They have many pieces, but they can act in simple ways. And he was showing that the paradigm that Haken showed for physical systems might also apply to sensory and motor systems. And the thing that occurred here is that when the system went through this phase transition, so there was a sharp change in behavior at a given value of a parameter, that a number of other properties were associated with this change if this self-organization and synergetics were happening. And they included things that the noise in the system increased. Sometimes there was hysteresis, so if you switched this way, you might get a different result going backwards, for example. So the shift wouldn't necessarily happen in the same place. And a number of other properties that would help you identify this as a synergistic system. And so that's one of the things that Scott Kelso has been doing in a number of different experimental paradigms. And this is really an example of bifurcations. And they were able to model this, again, based on the initial physics, as a potential well where the finger is doing one thing if it's in one part of the well and doing something different if it's in another part. And as the parameter changes, the shape of the well changes. So we start with two nice defined wells, but then one of them disappears. And so the system has shifted from having two capabilities of your left and right finger moving separately to only having one capability of having both fingers move together. What this means, and I think the take-home lesson here for biology, is we're used to thinking that, as I said last time, that if you go into the incubator and take out a 1-inch or 2.5 centimeter cell dish with cells growing in it, and you plop some chemical in it, that if you get a certain effect when you do that experiment on Tuesday, you'd like to think that if you take the cells growing next to them and plop in roughly the same amount of the same chemical the next day, and you can never weigh out the chemicals exactly the same either, so they're always going to be slightly different, you would like to think you would get the same effect. And that intuition is based on our experience with linear systems. And it's not necessarily true for nonlinear systems, and it's not at all true for chaotic systems. So we could actually run an experiment where we have a bifurcation where we vary the amount in a very small way and get a marked change in behavior in the system. And this is a lesson that people, are really not used to hearing in a way, because we presume if we set up the experiment in roughly the same way and run it, we're going to get roughly the same response. And that's not necessarily true. And also, in determining whether a system is chaotic or not, one of the ways to do it, or to look for a deterministic system, is that if we have a mathematical model, for example, a self-organizing synergistic model, and we have an experiment, we can predict the bifurcations from the model as Marcus and Hess did for glycolysis, or as Kelso did for the finger tapping, and we can see if the experiment runs through those bifurcations, and if they match, then we know we have a deterministic system, and we can understand more properties of that system. Or we could look at the phase space of the experimental data. So if we have the experimental data and we look at the phase space tells us if the data was generated by a random or deterministic mechanism. Because if the phase space is low, if the attractor in the phase space is low, then we have a deterministic system, and we can tell the number of variables from the dimension of the attractor. And if the phase space is very large, if it's infinite dimensional, then we know that the mechanism that generated the experimental data was due to chance. And let me give you some examples of places where this has been used. I mean, if you think about it, this has really been a revolutionary idea in biology, because we're always used to seeing results in biology that are very variable. And we're used to having, for example, the same drug produce different effects in different people. We're used to doing experiments on different animals and having the results very different on each animal. And people thought that there was some intrinsic variability in the biology, and so that each time you did something, the body responded differently, or each individual responded differently. And suddenly this is saying that maybe those differences in response are not because there's tremendous variability in biological or living things, but it could be due to chaos. So people have been very excited to apply a lot of these ideas to different systems, to analyze them, to see whether the variations that people see are due to randomness or due to chaos. So there are two parts to this. The first part is the good news. And the good news is that whenever we saw a complicated looking signal, we could now do this analysis, which will tell us whether it's really random, that is stochastic, or whether there's an underlying determinism process in what's going on here. And since, of course, there's good news, there's, of course, going to be bad news. And we'll get to the bad news later. So I didn't ask you if you wanted to hear the good news first or the bad news first. And let me show you examples where people have applied this. For example, people get measles each year in New York City. A lot of diseases that people get in the winter are not due to the fact that their immune system is weakened in the winter, but the fact that they're all together in rooms. And so a lot of diseases spread faster under those circumstances than in other cases. And that is probably the reason why measles or other diseases spread more during the winter. What's plotted here is a function of time with the number of measles cases in New York City. And you can see there are fluctuations which are mostly in the winter, So that's why this has a somewhat regular looking pattern. But you can see some years a lot of people get measles. In other years, not so many people get measles. So there's a tremendous variation. And the same thing is true here for the chickenpox. So again, we can see that there's an annual cycle, again, higher in the winter. But some years, a lot of people get it, and some years, not so many people get chickenpox. What the question here is are these variations year to year really random or not? And what they've done here is they've embedded the data in a phase space and determined the dimensionality of that phase space. And this shows some of those results. This is for Copenhagen. This is the correct spelling of Copenhagen in Danish, including the vowel. I was once in trouble, I think it was in Scandinavia, trying to find someone's name in a phone book because their name began with one of these vowels, and I didn't realize that the way you alphabetize things when you have vowels is that those go after Z's. So I couldn't find their name in the phone book because I thought it started with an A, but it actually started with a letter that came after Z, so it took a little while to figure that out. So alphabetically, these are done. This would not be an O, this would be after. In any case, Milwaukee doesn't have anything like that. Is this the correct spelling for Milwaukee, or is there too many E's? That's correct. And St. Louis and New York. And what this group did was to determine the fractal dimension of the attractors in the phase space. Now, what their conclusion was from this, they had very little data. So actually, these numbers for the chickenpox, which are close to 3, might, as far as they can tell, really be infinite. But they thought the measles had a lower fractal dimension. So they thought there was some deterministic pattern in how the measles were being spread, but not necessarily how chickenpox was being spread. And there's a standard model for the spread of epidemics called the SEER model, where you look at how many people are susceptible, that is, haven't caught the disease yet, how many people are exposed but haven't developed the disease, how many people are infectious and can spread the disease, and how many people have recovered. And these are again coupled by a set of ordinary nonlinear differential first-order equations. And from varying the parameters in this model, this suggested that the measles was really the variations in the measles were really due to variations in the parameter of this model rather than some intrinsic stochastic or random event. So they thought that the measles was more chaotic and the chickenpox looked more like a noisy, that is, a stochastic annual cycle. But again, there's a relatively limited amount of data, so these conclusions aren't absolute. Another example is in the beating of the heart. We described before that the heart beats each beat separately, but it gets organized overall into a wave of activity. And this shows voltage being recorded from the heart in an electrocardiogram. And we can turn that into a phase space. When the heart is beating irregularly, this is a recording made from a very irregular beating heart, it's gone into fibrillation, which is not good. And in this case, the phase space here was very high dimensional. So a number of people have been looking at the dimensionality of the phase space to see if they can, first of all, identify or characterize different arrhythmias and also see if there's some hint in the phase space before you get into here, so it would have some predictive value. And different people have found different results for this. So let me just leave it at that. Let me go back a second, because there's actually one issue here which really is interesting, or a couple issues that are interesting. And this kind of shows where the mathematics is. If we have a heart beating, so we're measuring the voltage coming out of the heart versus time, it will look something like this. Actually, that's pretty good. So each of these bumps means something in terms of the heart. This is called the QRS complex. So these are indicated by R's. So the time between the heartbeat is very often called the RR interval. And I always forget whether these are capital R's or lowercase r's. I mean, I'm sure I would do this in front of an audience that would know, and they would laugh at me, and then I'd be in a lot of trouble. But I don't remember. In my favorite book, probably is my favorite book, which is The Little Prince by Saint-Exupéry, which may or may not be spelt that way. It is? No. How is it spelled?
I think it's with double E, a closer complex.
At the beginning, a double E?
No, at the end.
E-E-X. I remember a Y rather than a double E. But it doesn't matter. In this book, there's, this is supposedly a children's book, but it's not a children's book at all. It's really a really tough, nasty book with a really sick ending. Not sick, but very tough ending, and which I won't give away. And I think Saint-Exupéry is actually one of the best writers of the 20th century, and is really interesting. And I was in a bookstore recently looking for something else where I came across a number of his books, and they're really excellent. He writes mostly essays and stuff. Wrote mostly essays and stuff. He disappeared during World War II flying an airplane in southern France, maybe the middle of France. But there's a really nice scene toward the beginning of this book where he has an astronomer present the discovery of asteroid, I think it's B612, and he has there's a picture of this guy presenting something on one of these big pads for some corporate presentation. And the astronomer was from Turkey, and so he's dressed in traditional Turkish dress. And so nobody believed what he had to say because he was dressed in this funny way. And this is actually a reference to Ataturk, who required people in Turkey to dress in Western dress and suits. And there's another picture of the same guy, but dressed in a suit with the same thing, board with the same stuff written on it. And this time he presented it in the Astronomical Congress the next year. And this time everyone believed it because he was wearing a suit. So that's the sort of things that are sort of in this book. So I'm going to get really into trouble if I don't figure out someday whether these are capital R's or lowercase r's. But here it doesn't matter, as long as I don't tell stories like that. So we now face an actually interesting issue in analyzing this data. If, in terms of this data, what we think is going on, or the thing that we think is important to measure, is just when the event happens. So we've started off here with a continuous process, a continuous recording of voltage. And if we only ask when it happens here in time, here, and here. And this is a different type of process, which is called a point process, because we have the points in time where things happen. Now, we can certainly measure the intervals between those points, but it turns out the mathematics of a process like this is different than that. And for a long time, the embedding theorems, remember what we're trying to do is to convert the time series of data into some geometric object and then study its topological properties. The theorems that allowed us to do this were derived from continuous data streams. So we could take this whole voltage as a function of time and create the object out of it. But those theorems were not designed to handle point processes. So how we handle data like this was a little bit unclear. A number of people, including Mingzu Ding and Jim Collins at Boston University, have shown recently over the last year or two that we can actually use methods similar to continuous data to handle these point processes, and those methods should work. But actually, some of the mathematics of doing this isn't proven. And it just goes to show that people are still actively doing the mathematics of these things. And a lot of issues come up in how we handle data. Maybe I'll say what the mathematical issue is, which is that if we have some function of time and we're going to convert this into a phase space, typically the real phase space that we want would consist of, for example, derivatives. So what we want to do is to pick points on this function so that they're far enough apart so we get a good estimate of the slope. If we pick the points too close, it's kind of hard to figure out the slope. And if we pick points too far apart, the slope is also not a good estimate. So how we convert things for this continuous process, there's certain rules. But if we don't have something that's continuous, we just have disconnected points, then we have no slope. So how do we derive this phase space? And that's what the mathematical issue here is. So it involves different properties between disconnected points and smooth functions. Another case where this comes into play on the difference between what I can call, accurately call, continuous functions, where the next value of the function depends on the previous values plus their derivatives, as opposed to a mapping or a discrete function, where the next value depends on the previous values at discrete points. For a continuous function, if we had a phase space where the functions crossed, we couldn't tell what direction they were going in, because if we're coming in here, do we come out there or come out here? So in a real phase space for continuous functions, if everything is cool, then the functions never cross. But when we have discrete functions, we have just points. So it's no sweat if the things cross, right? Because we're very unlikely that the 2 points are going to fall on each other. So how trajectories for, so these trajectories for discrete systems have different properties than this, and continuous systems. And one of the consequences of this is that the original work of Poincaré, which was also led to a theorem which is now called the Poincaré-Bendixson theorem, is that in order to get chaos in a continuous system, we need 3 or more independent variables. But for a discrete system, we can have chaos in 1 or more variable. So this means if we have a continuous system that only has 2 variables, it can never be chaotic. And the proof of this is called the Poincaré-Bendixson theorem. And Poincaré has accents on his E's, but not Bendixson. And I don't know who Bendixson is. There's a numerical method called the Newton-Raphson method, and everybody I know knows who Newton is, and nobody I know knows who Raphson is. So, but I think Bendixson is a modern mathematician. Okay, so it's interesting that these rather subtle mathematical points can lead to really big change in the behavior of systems. So you really do have to go back to what the underlying mathematics here is, and that's really interesting. That's something that shouldn't make much of a point. I mean, whenever we collect data, we always collect points, right? We never collect things. We can never measure things infinitely fast. We're always doing something like this. And actually, when we're doing this, we're not doing the mathematics that's really differentiable. So all of the mathematics we use is meant to mimic this, and we think for most cases that this is going to be okay, but we can see we can get into situations where actually it's not okay, and that has a real consequence as to whether the system could, in principle, be chaotic or not. So it has real practical consequences. Okay. Oh. I just had a message that appeared on the screen that says the system will shut down in 1 hour. It would be nice if it shut down now and I could restart it, but the idea that it's going to shut down in one hour is kind of scarier. And then it warned me it would do that. So another example of data that looks random is what comes out of our brains, which is a little bit scary if you think about it. You would think that what comes out of your brain should not necessarily look so random, but apparently it does. And so maybe it's not really random. Maybe there's something going on here. Now this is really more complicated than that, because this data recorded by electrodes sitting on your hair is really an average of many different things happening at once. So maybe it looks random because you're averaging over many things at once. So, I mean, for some reason, I picture a very graphic example of a number of different CDs players set up with very big speakers, and one of them is playing Mozart, and the other is playing Savage Garden, and another one is playing Paco Bell, and another one is playing Barry Manilow. And if they're all equally loud, then it might sound very disharmonious. And I can picture this in a room with carpeting and the lights turned down there. But that's kind of what we're recording here in even a much more extreme way, because we're really averaging over many different signals. And why this has this character of looking random may have to do with the fact that we're averaging only currents near the top of the brain, and that we're averaging over a large number of cells. The issue is there additional data that's contained in here? And so people have done these embeddings, again, plotting the voltage now against the voltage delayed some amount to tell whether there's an underlying order here. And some of the analysis, for example, has found low-dimensional attractors, dimension 3 or 4 or 6 or 8, and other data hasn't. And this is a short list of a number of people who have done this. This is not really an up-to-date list, but to just give you a sense of the wide variety of people in different places who are doing this. So this is a group in Philadelphia, and this is a group in Brussels, and this is a group in Beijing. So it really covers a lot of different places. Now, there's a lot of problems with this data. Different groups have found very different dimensions under the same experimental conditions. I would think that a lot of people are beginning to think now that a lot of these dimensions from the attractors are probably not real. That this analysis has been done on a limited amount of data, and the finding of low-dimensional attractors from that data may be more due to the fact that there's not enough data rather than these attractors are real. So I would say a lot of this work now is at least not necessary, not that it's wrong, but I think there's a little more skepticism about this than there used to be. If there is anything that comes out of this, it seems to be that you get lower dimensional attractors from this data. That means fewer things are going on and it's more deterministic. When you're really in some odd state, like you're meditating, this Qigong is sort of a yoga-like Zen martial arts form of meditation, which is very popular in China. If you're in epilepsy, and so this is actually work done by people in Beijing, because there are this is something that a lot of Chinese people are into. And even if it was a small percentage, there would be a lot, because there are a lot of people in China. And in epilepsy, in a petit mal seizure, the brain waves are very regular. Or when the brain isn't working at all because it's been infected by a virus, which is basically distorted proteins and has killed a lot of nerve cells, then this reminds me of a story that I'm not going to tell. Basically here, the brain is functioning in a very regular way, which doesn't look at all like this very irregular pattern that I have here, but looks very periodic. And the person is not really conscious about what's happening around them in all three of these states. As you go into quiet sleep and quiet awake with your eyes closed, the dimensionality of this signal increases, going up to a mental task. The usual mental task done in these experiments is to start at 100 and count backwards by sevens, which gets difficult by 2 or 3 times, okay? So it's like 93 and then 86, 79, no, yes, no, 72, okay, 60 something. So the idea here is that the dimensionality represents the difficulty of the task, maybe. I've done this in terms of ion channels. That is one of the things we've been trying to investigate in terms of how ion channels work is when we have an ion channel protein. Let me say a little bit more about this, but not too much more. If we measure the current going through a single ion channel in a cell versus time, and we do this by putting a micropipette, which has an opening here somewhat enlarged for you of about a micron, and then we seal a piece of cell in here. So it has typically one ion channel. And then we measure the current flowing through here. So this little symbol represents about $150,000 worth of equipment. So it's bigger than this. The currents look something like this. So the channel is either, let's say this is zero current, is either open, which I usually write as green, which I don't have a green, so we'll call this open, is an open state. In the closed state, I write as closed in red. I don't know if this helps or not. I thought picking red and green for open and closed in terms of traffic lights actually it was a good mnemonic. So this channel, this protein, shifts in between states that are either open to the flow of ions and we see a current, or it's close to the flow of ions. And the question is, how does this protein, what happens when this protein goes from one of these shapes, which in essence has a hole through it, to another shape which is closed? And what is that transformation telling us? How does this happen? Is this a random event? Is this a deterministic event? And so I've been using chaos to look at that, to analyze this data to tell whether the channel, whether the transitions in the channel are due to a random or deterministic event. And what it would mean is that in the usual picture of how we think molecules switch from one state to another, when we have a small molecule, it basically has different energies. And it's in a state, and every once in a while a little heat comes along, and it goes click and moves to another state. But this issue of whether molecules that are big, like proteins, work like this has not really been addressed. And what might be going on in terms of the channels is that if this is a real large molecule, it is a real large molecule, about 1,000 amino acids. So it's big enough to, in essence, act like a little miniature mechanical system, as if it consists of balls and springs, and it's sort of shaking. And if it can do that, if it acts like a mechanical object and has properties that are related to real mechanical features like stiffness and springs and squishiness, and then what may be happening is that motions may start in the molecule and amplify themselves and cause it to switch from one three-dimensional structure to another. And we're trying to use these methods to see which of these two processes is more important in how channel works. So far I've been discussing the good news, which is in principle you can tell if the data was generated by a random or deterministic system. And based on the good news, that's how we're analyzing our data to tell whether the switching from a channel from one state to another is really random or deterministic. And the answer we got from analyzing our data was that we couldn't tell. So now we get from the good news to the bad news, which is that it's not always easy or simple to use these methods. So let me just review some of the problems in these methods to give you a sense of why it's not so easy. First of all, it's not clear how much data we need in order to do this analysis, but it may be very large. In one of the other of these, I don't know if it's on the PowerPoint slides or not, but in the transparencies, it says here you need a humongous amount of data in order to analyze this. And I remember that because I didn't know how to spell humongous, and it's not on my word processor. But for some reason, I was surrounded by humongousness, I guess. That week I was trying to do the transparency. And there was an article in the New York Times that had the word humongous in it, so that was one spelling, although I think it had another U that it seemed different. Humongous is a very California word. When you're in New York, it's very hard to figure out how to spell that. And Another thing that came up was, and we always forget this, we had a discussion last time, it's the second Road Warrior movie that has Humongous, who's the guy with the, in the Mel Gibson movies. He plays the big bad guy in the movie, and he's got a mask, a hockey mask, and the name of that character is Humongous. In the Road Warrior movies. It's the second one, I think. So in any case, this was before VCRs, so the movie was on TV that weekend that I was preparing the transparency. So I had to watch the movie all the way through to get to the credits and not like fall asleep or be off in the wrong room to see how they spelled. And the movie is made in Australia. They spelled Humongous differently than the New York Times did. So, but I think neither of those corresponded to the spellings I had on the transparency. So here the word humongous has apparently been replaced by very large, unless it appears on the next slide. And large may mean 10 to the D. And the basic here is that the sampling rate in some way must cover the attractor evenly. And if we sample too often, we only see the trajectories. If we sample rarely, we don't see the attractor at all. So let me say that if we have an attractor like the Lorentz attractor, if we sample too frequently, then it just looks one-dimensional. And if we just sample here and then go over there and there, then it looks like it's all spread out. It looks like it's very high-dimensional. So how we do the sampling is very important. So first of all, we need enough data to get around each trajectory, at least once, and then go around several trajectories. So that adds up to a lot of data. And whether we can collect that data in a physiological system where the system remains constant may be difficult. Second of all, in doing these lag plots that we've been doing, we need to use delta t. This is the issue in terms of doing the derivatives. As I said, if this is too small, the variable doesn't change enough. If it's too long, the variable changes too much. And as I was also saying before, the embedding theorems apply to some cases, but not to all cases. So we know they apply for continuous smooth time series. We know they don't apply to discrete time series, although people are working on that now. And we know they don't apply to fractal time series. So let me sort of summarize in terms, remember the whole point of this is the embedding theorems tell us how to go from a time series to an object. The theorems are well understood if the functions are continuous and smooth. Smooth technically means that the second derivatives exist. If the series are discrete, we have some theorems to tell us what goes on. If the time series is fractal, we have nothing to tell us whether it works or not. Okay? So this just gives you a sense of where the mathematical certainty or uncertainty is in handling this data. There's really a range of different things. We understand how to handle some data well. Other data we can understand a little bit. In some data we have no idea what to do. Well, this is good because it means people are currently developing mathematics of this. But I just want to give you a sense that it's not that there's a series of methods and we can handle everything. The other thing, which is a point I've emphasized before, is that real experiments and real data are really driving new mathematics. Because since we have data that has properties unlike the mathematical, unlike the assumptions and the theorems, then it creates interesting new approaches for developing new theorems that deal with cases that haven't previously been done. So it really is useful, I think, because it will drive new mathematics. Okay. And this shows each of that in some detail. For example, how many values in the time series do we need to evaluate the dimension? And what I've gone here is through a number of articles that have been published and showed how many values they said you needed. And each of these things is based on a specific argument. And each of these articles justifies their argument in terms of a numerical simulation. So there's some work to back up. So each of these are right. And if we look for a d-dimensional attractor, Smith et al. Said we actually need 42 to the d points, which if d was 6, this works out to 5,000 million billion. Is that right? Is this 5 billion? I mean, American billions, not British billions, which are bigger. British billions are 1,000,000 millions, whereas our billions are 1,000 millions. And the world is roughly equally divided between people who use British-type billions and we who use American billions. So Germans are American billions, but I don't remember what French are. It's hard to believe the French and the British would do the same thing, but anything, they don't. Okay. Very often, especially in textbooks that are published in Britain for the American market, although they may use British spellings, they'll very often say 1,000 millions. Or if they're using billions, they won't say billion, they'll say something else in order to prevent confusion with the American market. So there's some sensitivity to this. So if we wanted to see a 6-dimensional attractor, Smith would say we need 5 billion data points in the time series. Wolf, who's in New York, his office is near where I used to live in New York, at Cooper Union, is 30 to the d points, which would be 700 million. Okay. And a different estimate may be 1,000,000 points. These guys, who I don't know where their office is, said 200,000 points for the same attractor. Let me actually give some hint as to what this is. This is, we want to sample each trajectory in the attractor and sample many trajectories. That's where these two come from. This is, if we're filling up a space, we need roughly 10 to the d points to fill a space. Here, if we're looking at determining the fractal dimension, that will be determined by the surface to area measurement ratio. I won't explain what that means, but that's what that's based on. This is based on a similar thing. Both of these are based on the fractal dimension depends on some scaling, and these are based on the scaling. Ding says we could do this with 1,000 points for a 6-dimensional attractor. This is also based on a similar argument, but fewer points. So you can see that these estimates range from 10 to 5 billion data points. So it's like real hard to decide how to do an experiment now because there's a tremendous range of data points. So, and it's not clear how much data we need. And this goes to the point I said before about picking the time lag. And this is actual data from the Lorentz system, computed as usual in BASIC. And if we plot the x value in the Lorentz system a little bit later against the x value now, they're always going to be the same, right? Because it's similar. Because if we wait on a very short time, whatever it was doing now, it's going to be doing the same thing a short time later. So if delta t is small, we always get the fact that the system is on a line. It's one-dimensional, and therefore deterministic. If we wait a very long time, then what happens now is not correlated with what happened a long time ago. And so we always get that the Lorentz system is high-dimensional and deterministic. It is only when we pick delta t just right, which is why I usually call this my Goldilocks transparency, because, right, okay. So that we actually properly reconstruct a Lorentz attractor. So in differential equations, which is most of the mathematical systems for which these were derived, we know how to pick delta t. But for real data, very often we're in this situation, and when delta t is small, it looks low-dimensional. When delta t is a little higher, it looks medium-dimensional. When delta t is very large, it looks high-dimensional. And which one of these three things gives us the correct dimension of the system is not clear. For ordinary differential equations, we can tell this is the right one because there are certain other properties related to correlation and mutual information and entropy that are special for this. But for real data, that's not necessarily the case. So creating the phase space has some problems. And lastly, this is what I was saying about these embedding theorems. Tockin's theorem, one of the embedding theorems, says that we have to have the system being smooth, and if it's not smooth, then it's not necessarily true that we get the right result from the theorem. And okay, and people are approaching this by trying some other methods of analyzing data. And maybe what we'll do is we'll take a 15-minute break, and I'll come to some ways that people are trying to handle this at 10:30.